Well, thank you for clicking on my video and welcome to my video. My name is John. I'm an advocate for patients with glioblastoma and other brain tumors, and I'm making this video to express the importance of saving tumor tissue frozen. The reason why is because there is a vaccine which might be available in a few months that has saved lives in clinical trials. Those patients with glioblastoma or anaplastic astrocytoma. And the reason why I'm doing this is because historically the medical community would be freezing, would be actually paraffinizing tumor tissue instead of freezing it. Paraffinized tissue is typically used for study for submission into clinical trials, but it can't be used for vaccine manufacturing. A couple of disclosures first. I don't have any medical education, any formal medical education. Everything is self-taught, and I also own shares in the company that owns Northwest Biotherapeutics. So what I'm going to hopefully do in this video is talk about what we've learned so far about this vaccine, its possible combinations, how the vaccine is made, why tumor tissue has to be frozen and can't be paraffinized, and what I think would be one of the reasons why I think that we should be off-labeling some of the immunotherapy combinations instead of going with a lengthy clinical trial. That's just me as an advocate, but it's more of an educated guess based on logic, not as much as it is on science. But when you consider what's at stake, everything kind of ties in together. But talk about the vaccine here. This is a screenshot from a press release and from a journal article from 2018, and it shows that the top 100 patients are doing very well. The phase 3 clinical trial is a 331 patient clinical trial. The 2008 data shows that the top 100 patients are living almost 6 years on average. The median overall survival from surgery is 58.4 months, and median overall survival simply means half the patients are alive and half the patients have moved on. And it does indicate that 8 of the 100 patients had only 8 of the patients had favorable status on key prognostic factors combined, which was younger than 50 years old. They had methylated gene status, and they had a full surgical removal of the tumor. So it's not like cherry-picked data. It's not like the patients were specifically picked during recruitment to meet these criteria. These patients came out successful because of the vaccine, is what they're saying. And here is two cells. They're both dendritic cells. The one on the left is a naive dendritic cell that hasn't been exposed to any cancer material. The one on the right is a antigen presenting cell. Antigens and receptors, they kind of are the same thing. The way that the vaccine works is the cell, so like the naive dendritic cell will be exposed to cancer material which we would have removed from the patient. So that's when we saved the frozen tumor tissue. We thaw it out in a 5-cycle freeze-thaw, freeze-thaw, freeze-thaw cycle. What that does is it removes the cell surface antigens off of the cancer cell. Those are all unique antigens. What we are now using as targets. And so once we take those antigens off the cancer cell that we've managed to keep alive by freezing it, we introduce it to the naive dendritic cell, which uptakes those antigens. And now what we have now is like a vaccine. The one on the right, we can posturize as that as being a vaccine. The dendritic cell then is injected back into the patient, into the bloodstream, or just into the intradermally, and it will then mingle with other white blood cells that are naive themselves as well. And those naive killer cells, white blood cells, will uptake what the dendritic cell is offering up, which is those antigens, those receptors or targets, and it will find the corresponding antigens or targets in the body, which would be at the tumor site. And that's what the white blood cells knows, is to go find the corresponding antigen. And we do have some information as far as what some of the characteristics are of the patients that are surviving the longest. And one of those characteristics is that they have been these patients that are living longer have been identified as being from the mesenchymal gene expression. And so I'll read from here. This is from an earlier phase trial, around 2015 if I recall. Patients whose tumors had a mesenchymal gene expression signatures exhibited increased survival during DC vaccination compared to historical controls of the same genetic subtype. In other words, if you look up mesenchymal gene expression, typically those journal articles will indicate that these patients had poor survival ratings, but that would have been because they would be corresponding to chemotherapy and radiation. There's a different element when we're using immunotherapy, obviously, and with these mesenchymal gene expressions, there's something about these mesenchymal gene expressions that we have to try and figure out what makes that mesenchymal gene expression such a type of patient that would be able to do so well with this vaccine. Okay, so now we know that the mesenchymal gene expression is the gene expression that does really well under DC vaccine therapy. So what will be good to know is what makes the mesenchymal gene expression different from the others. And to start, we would say what other gene expressions are there, and this chart shows that there are 4. There's proneural, mesenchymal, classical, neural. And I do apologize on this being a little bit dry, but crash course will basically show you that let's not try and understand what the DNA strand and what the ladder of a DNA strand, like what it all means. Like, just for all intensive purposes, let's just understand that there's 4 gene expressions, and a patient will probably have 1 in 4 chances of being in the mesenchymal gene expression. And then to kind of fast forward, what makes one genetic expression different from the rest? So what makes a mesenchymal gene expressed patient different from a classical or a neural? And so what I've basically kind of put it on myself is that I've tried to find out what some of the larger defining differentiating factors are in those genetic expressions, because that might show you why these patients are living longer. So I've got an arrow here on the screen. I'll get to that later, but what's good to know, that what I find, and this could be subject to debate, that some of the larger differentiating characteristics of these 4 genetic expressions are, I found 4. One of them is tumor cell type. So if a patient has NF1 cells or a lot of EGFR expressed cells or PDGFRA, all the scientists will know, like just by looking at them and by staining a slide, what type of cell is in that cluster, and that would point them to this specific genetic expression. Okay, so that's one. That's like the cell type. Another one is immunosuppression. Immunosuppression, which this chart discussed, is a molecule or different types of molecules that a tumor will release to neutralize a white blood cell or a vaccine. And a white blood cell is a vaccine. So you can see on the left, from VEGF down to CTLA4, these are all molecules that are either on a white blood cell or on the tumor, and these are immunosuppressants, which interfere with the antigen to receptor handshake, which is what is required to create tumor cell death. They're just getting in the way of that mechanism of action of antigen to receptor, and the scientists have been able to quantify how much of these immunosuppressants exist based on, and then they assign it to a specific genetic expression. So for example, a mesenchymal gene expressed tumor will have the highest amount of CTLA4 in their compared to the other genetic expressions. So a proneural patient might have 9% of their cells expressing CTLA4, for example. Another way to differentiate genetic expressions is tumor infiltrating lymphocytes, which was on my other slide, showing that mesenchymal patients did have a lot of tumor infiltrating lymphocytes. Tumor infiltrating lymphocytes are basically killer white blood cells that make its way into the tumor. So if a scientist would to resect a tumor and look at it, the inside of it under a microscope, they can see, well, this cell has a lot of white blood cells, killer white blood cells that made their way into the tumor. So probably we're going to say that's a mesenchymal expressed tumor. Or they can look at other tumors, like so maybe there's not a lot of white blood cells, maybe that's a different genetic expressed tumor. Another big differentiating factor to classify genetic expressed tumors are T regulatory cells. So in the tumor microenvironment, they will have T regulatory cells, which the body has naturally to tell the immune system to take it easy, to not go around killing every cell in sight. We kind of need this. This phenomenon is studied a lot with organ transplants. When you have an organ transplant, you will have you will have a mechanism of action where the body will perceive something foreign in the body and will turn against it. So it'll turn against the liver or a kidney, so on and so forth. And doctors are looking at ways to minimize this mechanism of action, and they've also been able to quantify the number of those cells in a certain cluster and point it towards a certain genetic expression. And so those are the four big ones. Okay, so in regards to the PD-1 status, the reason why I highlighted it or pointed towards it is because one of the biggest anomalies for mesenchymal gene expression is that they have the lowest amounts of PD-1, but they have the highest amounts of everything else. So how important is PD-1 in cancer terms? Is that something that scientists are studying? Is that something that is coming up in discussions with other cancers? Yes, it is. So this study tested Keytruda, which is also known as pembrolizumab, on non-small cell lung cancer. And Keytruda targets the PD-L1 PD-1 handshake, which is what the tumor uses to neutralize a white blood cell. As it relates to PD-1 PD-L1, this study was able to provide a median overall survival of 30 months to patients taking pembrolizumab. And in patients with just chemotherapy, their median overall survival was only 14.2 months. So there was a benefit, and this study did show that patients who quantifiably had higher levels of this vaccine, or sorry, not of the vaccine, of the PD-L1 scores, that they benefited from this checkpoint inhibitor. And as my previous slide did show, that I was pointing to mesenchymal gene expressed patients with low levels of PD-1. The way PD-L1 and PD-1 work together is, you can see on the left, the diagram on the left is a tumor cell and a T cell. The T cell, what it wants to do is use a T cell receptor to antigen handshake, which would kill the tumor cell. That's the idea, but the tumor's defense is this PD-L1 PD-1 mechanism of action, which intercepts that handshake. But on the right, we have anti-PD-L1 and anti-PD-1 course therapy. And once we implement those, once we block that handshake with either PD-L1 or PD-1 checkpoint inhibitors, then that will allow that antigen to receptor handshake, which initiates the tumor cell death. Although I'm quite confident that my logic does have some substance to it. Maybe a counter argument could be that you could see in the proneural subgroup, there's the PD-L1 numbers are 0, meaning that there's absolutely no defense on the tumor cell. And maybe, so why would they not be doing so well instead of mesenchymal with DCVax vaccination for GBM patients? And the only thing I can say is that sometimes the readings can vary. So this is a separate study. It shows that there are PD-L1 expressions on proneural glioblastomas. This was from 2010. So I don't know if that just qualifies as maybe like trace elements or not, or what the argument there is. I do also know that sometimes a glioblastoma sample can express levels from both subgroups, so it could be partially maybe mesenchymal and partially proneural. There are some ranges involved, so I don't think it takes away necessarily from the argument that that's what potentially why mesenchymal patients do so well, but I do think that there's more there that scientists are going to lead us towards. There is also one thing I did want to bring to the attention, that seeing that a tumor can present more than one immunosuppression, they can indicate PD-L1 and CTLA4, so on and so forth. But that also gives an opportunity to maybe use more than one checkpoint inhibitor, which would which again targets those immune suppressor genes. So I know of a mouse study that was done years ago on and these mice were given glioblastoma tumors, and they were each given a combination of checkpoint inhibitors that targeted CTLA4, PD1, and I believe it was the galectin-3, the LAG-3, and all the tumors. All the mice survived at the end of the experiment, and the control mice did not survive. So that's something to consider. Like, there's you can throw more immunotherapy at a patient, theoretically at least, than just one, like a vaccine or a checkpoint inhibitor. Ideally, like if you're not worried about toxicity, a glioblastoma patient may want to consider multiple checkpoint inhibitors along with the a vaccine therapy such as DCVax. So that mouse study is accessible online. It's on NIH.com's website or NIH.gov's website. And so, sort of recalibrating, going to a different study now, something that's super important, something that I thought that would be very pertinent to, and what may simply be added to every treatment protocol if DCVax is approved, is this study, which includes Imiquimod to DC vaccination. Imiquimod is a FDA-approved drug for skin cancer, and what it does, it's like a cream. It's applied to the skin cancer, and what it does is it brings the white blood cells up to that area of application, and there's no freezing or anything like that. It's just bringing white blood cells to the area of application. And so the way a neuro-oncologist who's treating a glioblastoma patient would use this, as they did in this study, was 24 hours before vaccination, they applied the Imiquimod to a patient's arm, and then the next day there'd be a concentration of white blood cells going to that area of application. From there, the dendritic cell vaccine would be injected into the patient intradermally, and when you're doing that, you have a higher concentration level of dendritic cells that will now be mingling with that higher level of white blood cells. So you're getting your vaccine, now it's mingling with a higher number of white blood cells. And what you're doing then is you're eliminating the kind of inefficiency when you need that interaction between the dendritic cell and the naive white blood cell. You're bringing that to the table immediately, and then they can go hunt for the tumor tissue with the corresponding antigen. That's what the Imiquimod does. And you can see, like, these patients are living 88.87 months, 91.3 months. And my other slide, these patients were living 40.5 months and still alive at the time of study, 59 months still alive, and this one patient was with the 40.5 months. They were given the Poly ICLC. That is it's got the same idea. It's just a different drug. What another thing that brought to my attention was that these patients were just given one dose of the vaccine. So in the clinical trial, they were given multiple injections. They were given ones, I think, 3 or 4 in the first 6 months, and then one every 3 months afterwards. And then here you're just getting one vaccine. That's what it says to me. You see their KPS score, which is their cognition skills, so on and so forth, like pre-vaccination. So like, how are their motor skills? How is their functioning? How do they have any neurological deficits? So if they have a high KPS, that means that they're doing well. Overall survival. HC type, that's histochemistry type. That's mesenchymal, proneural. Star must be like a combination of genetic expressions. That's your dose. That's not 10 to the power of 6. That's the number of dendritic cells that are given to the patient. I know that's also a higher number of dose compared to other maybe private pharmaceutical companies that have approved therapies in other countries. They don't give you as much dendritic cells per dose, I believe. So I think Northwest Biotherapeutics was going between 5 to 10 million. I know other companies were giving like 1 million. At least that was a few years ago. And another thing with Northwest Biotherapeutics, they also add a compound called interleukin 4 or interleukin 12. I think it's interleukin 4. And what that is it's a protein that helps the dendritic cell mature more efficiently. So when a dendritic cell is going to be in attempting to mingle with white blood cells and share that antigen information, it's going to be doing so more efficiently instead of it being in a confused manner. And that was discussed at the ASCO presentation, which is the American Society for Clinical Oncology. It's a big conference that's run in the States every year to discuss developments in the cancer community theater. And then there's other indication, or there's other treatments that the patients were given pre-vaccination. So temozolomide is a chemotherapy, as you know. And yeah, so like you can really tell how these patients are living longer with the combination of Imiquimod and DCVax. And that's DCVax and Imiquimod and mesenchymal gene expressions, and potentially how checkpoint inhibitors can replicate the tumor environment as far as expanding the survivor group past mesenchymal. The Imiquimod study is super important because, again, they're only getting one dose of the vaccine, if I'm reading that correctly, and that actually has happened in other studies as well. They have a vaccine using the same platform for other solid tumors, for like lung cancer, pancreatic cancer, and what they found is that when they injected the tumor site with DCVax, that if there was a distant tumor, a metastatic tumor, they found that shrunk as well. That had receded, and so that goes to show you how potentially powerful this vaccine actually is. And on the other side, if you're trying to plan your treatment plan with ahead of time, pre-approval, there's things that patients do have to actually, understand as well, as, or not understand, but to incorporate into their decision making is like there's several contingencies. This is all contingent that DCVax is approved. It's contingent that patients can get a hold of a biobank that can freeze their tumor tissue. If, for example, I'll just throw a number out there, maybe only 50% of hospitals have cryopreservation chambers ready for their patients. However, I do know that there's private biobanks like StoreMyTumor.com. They can provide frozen tumor preservation kits. They just need advance warning. You can contact the company before your surgery, and they can provide that hospital with the cryopreservation chamber and have it stored for you. And they actually work internationally as well. So that is a contingency that that process is complete. Another contingency is that the patient will be able to survive to the point where it gets approved. So maybe a patient wants to pursue ketogenic therapy. I can't really ketogenic diet or CBD THC. There was a clinical trial that was done in the UK which boosted survival from 369 days to 662 days in a 21 patient study group. I know that's a contentious issue in some areas because maybe medical marijuana is not approved yet. There's some backlash probably. I think I would consider it. I don't think it's been studied to the point where, so let's say like you are in, I know like for chemotherapy and bevacizumab, which is Avastin, some of these medicines, they'll actually change a tumor's landscape. So that means like if a patient was originally rich with NF1 cells and PDGFRA cells, then they're given chemotherapy radiation that will change the tumor landscape, the tumor profile, to potentially other cell types. In other words, other antigens. So if a patient had a surgery and they incorporated more medicine after surgery, and then that could change again, like I said, like the tumor profile, the tumor landscape. But when you're going to be getting a vaccine put together, and then when it's injected into the patient, it's going to be targeting those original cells. Now, I don't know how much of a change would take place of those. I don't know how much of a change would take place, but at the same time, you also have to think of getting to the point of survival. I know the vaccine has worked on patients that were in a who are progressing. So, you know, like say like hospice alarm bells are ringing. Depends on the patient's wishes. That depends on the patient's wishes, but if you do factor everything in, that you could maybe consider that there's still chance, there's still hope. If the science community isn't looking at CBD THC as part of standard of care, that would mean that the science community would say we've done everything we can do. But they're saying that because they don't know, they're not incorporating a 21 patient clinical trial of CBD THC into their discussion. So one thing I do like about CBD THC is that you know, it gets through the blood-brain barrier, and you don't need surgery for it, so it's pretty safe. It's quite effective. The way it works is that THC will actually bind to the cell, the tumor cell, initiate apoptosis. The way CBD works is CBD will make its way into the tumor cell and degenerate the cell wall through oxidative stress. And so that's more of a roundabout way of killing the tumor cell. Given the number of CB1 receptors in the brain and the central nervous system, it does indicate that THC could potentially be more effective at killing cancer than CBD in the glioblastoma. For other cancers, it might be different because of the cell types. But that's trying to get you from surgery to extended care. There's another contingency. You need to find a doctor that would be okay with prescribing off-label medicine. You need to find a doctor that. So let's say DCVax is approved. You have to find a doctor that would be okay with off-labeling a combination of other drugs. So find someone who's open to that, and then you'd have to just keep an eye on toxicity and things like that. As far as freezing tumor tissue, like if you were to look into clinical trials, so with respect to considering whether or not a patient would want to freeze tumor tissue, I did a search on phase 3 clinical trials in Canada, and there's 2 clinical trials for patients that are for clinical trials that are recruiting or not yet recruiting, so they're in the planning stage. And one of the studies was for intraoperative radiation, which is radiation when a patient is under surgery. And I'm not too sure about the process, like what the goal is, if it's for extended survival or complete response, like complete survival. I don't know what it is. The other study is for an investigational drug, and it has made its way through to phase 3, so it does have a chance. I just with the failures of, I hate to say, with the failures of ABT 414, TOCA, ICT 107, and rindopepimut, like these all made their way to phase 3, but then they all failed. I think they were in like different stages of cancer, whether it's newly diagnosed or recurrent, so they could be trying for the alternate version of glioblastoma. But I wonder if you did want to try this investigational drug, would it be possible to request that you keep some of your tumor tissue frozen? I don't know. But these actual studies are only available in like BC, Ontario, Quebec, Manitoba, and Saskatchewan. So Alberta and the Atlantic provinces, you don't have access to any of these studies. There might be like smaller studies that a university or hospital may be just trying to put together to offer something and to extend survival. But if they're not, then the patient really theoretically would have nothing to lose in order when they freeze their tumor tissue, if they're in those areas. And then in the States, there's definitely a lot more. Like if you're in the States, this is a map. I just arbitrarily picked vaccines in any stage for patients with glioblastoma. In reality, like you could go on Optune. Optune has a pretty good success rate for patients. That's the electromagnetic fields, or TTF. They're just it's a skull cap, a patient, not a skull cap, but it's like it's a cap that a patient wears, and electronic fields are sent, which inhibit growth of the tumor tissue. And I think they have like a 3-year median overall survival in a significant portion of their patient population. And then there's but here this map is showing all the vaccine options available, and there's absolutely, I don't think there's any there's none in Canada. But these vaccine therapies might be beneficial to patients as well, especially if they're not wanting to risk waiting for DCVax to be approved because it's been way too long. And if I don't know what the medical system is like in the States at all, what the copay is and things like that. So it's not my understanding exactly of what it is, but just to show you that there is more available in the States. I know there's like the Midwest and the Central America there's nothing available in terms of vaccines, but so let's say, for example, a patient is in like Iowa. I don't think Iowa it has any vaccines there. Let's say they're in Iowa or Wisconsin. Then they have to decide, I guess, do they want a clinical trial that might be investigational, or do you want to try and go for DCVax and have a potential approved product there? It does depend on I guess what all their homework would be put together.